I suppose the first thing I'm going to talk about very domain specific here, and that is basically in life sciences. Bioinformatics is where we, oops, I just spelled, right, you get to see everything very quickly. Bioinformatics is how we do informatics or how we do data science on biological data. And that's the field that I work in, and I specifically work in cancer. So how can we actually understand the pinnings of cancer to treat patients and save lives? That's where I work. And software is critical for that. And these are the two people that are here from my project today. Maria Doyle is the global community manager that connects our global community of developers. Our users, we have over 1 million individual downloads every year. Those are unique IPs, not joint IPs. And we have a community of over 2,000 developers. And this is what we're trying to do. We're trying to understand the DNA in every single cell. What causes disease? What is life itself? It's a pretty big question. And the technology to do that has changed dramatically. If you think of maybe way back in the day when you used a paper map to using your phone, that similar technology change has happened in life sciences. We have the genome, but we don't just have the genome. We have the technology, and I keep breaking technology. What happened? Okay, so we have the technology to look at cells on a slide and at every single gene that's being expressed in that cell, at every single protein, at the entire DNA of every single cell. One piece of data of a tiny, tiny piece of tumor tissue will contain tens of thousands of cells, millions of data points. And I would stop trying to break things. So we generate very large volumes of very complex data, and we need fast-friendly tools that are actually accessible to biologists. What we do impacts medicine, our understanding of biodiversity, our understanding of agriculture, marine plants, pathogens, microbiomes, how you live today. And understanding disease. What are the biomarkers, what are the blood biomarkers that we use in the clinic to detect disease, and new therapeutic targets underpinning pharma. And our success is actually indicated by 80% of new cancer medicines are molecularly targeted. So understanding how we actually analyze genetic data, the science in the universities that connect to the industry allow this to happen. And increasingly the data that we use is much more complex. So Bioconductor was established over 25 years ago, and it's a statistical package, a statistical suite of thousands of packages that support the analysis of biological data. It was written in R. It was originally founded by Robert Gentleman, who also wrote R itself. And then this was wrote when he was in Harvard. And I joined soon after that. And basically, there's a number of us that are on the governance board of Bioconductor. But what we do is we connect many, many different languages, underlying languages like C and Fortran, Python, Julia. It is not just R. It is that the statisticians and the data sciences use R, and therefore everything we wrap is in R. We are open source, open development. We're now in version 3.2. It's community driven. We have over 1 million downloads a year. And in a survey, actually, Kate was here yesterday. I presume she's not here today, is she? From CZI, and when they actually did a survey of the entire published literature to identify what were the most impactful open source software in science, 4 of the top 10 most mentioned software in the entire life science published literature were in Bioconductor. But we face a lot of challenges in building infrastructure that works. How can we have scientific methods that are sufficiently reproducible that they can actually be brought into regulatory affairs and be used when approving medicines, that they can be the basis of clinical trials? The data is very large scale. We have a very complex hardware landscape where we connect a lot of different technologies in the lab. We need to retain talent and institutional knowledge. And we manage this through Bioconductor. And the reasons why: we have a very, very strong governance, and we have a very strong maintenance system. And I'm going to talk about why we have become like a de facto place for publishing new methods in the biological sciences. We have a strong versioning system and a very regular release system. Time and time again, I've heard from pharma and people working in regulatory affairs that they can't just use code that's on GitHub because they don't know if it's going to be there in a year's time. They have to have projects that they rely on. And within Bioconductor, what we actually do is we do daily checks of every single package that's in our ecosystem. And we also have built a very, very strong community. We have strong codes of conduct. We have lots of languages represented and lots of technical backgrounds. So a little bit more detail. It's a package repository with a rigorous review process. Everything that comes into the repository from various different GitHubs, we actually do a review not just of the code, which is manually and automatically checked. We check that there's standardized data structures. We have really rigorous documentation requirements. Everything requires vignettes and tutorials. We have multiple books that are open source books written in Markdown. And we were fair before we're fair, so everything is findable, accessible, interoperable, and reusable. And also we're very much working within the global life sciences and health open source community, and there's a lot of projects in that space. Other projects we work with are Galaxy. We work with a lot with different Python communities, for example, SCVerse. I also lead Odyssey Ireland, which is the OMOP Common Data Model Standards for connecting health data worldwide. We took part in a lot of studies during COVID, one of which was over 1 million patients were looked at. We refuted the hydroxychloroquine erythromycin claims, and our paper was actually the basis of both the EMA and the FDA not moving ahead with that. And I also, as everybody has mentioned, I think the word that we've mentioned time and time again is infrastructure. And Elixir is the life science data infrastructure that connects 26 European countries. And because it's so critically important, and there's gaps in what we have here, I'm also head of Elixir Ireland. So, my last slide. How can what we have done help you, but also how can you help us? We have learned how to build a global community. We have an extensive Train the Trainer program. We have outreach in Africa. We have conferences on four continents. We have rigor, reproducibility, and robustness in our software through our quality check systems. And we have software that actually scales to very, very large data, multimodal data, and increasingly developing that for federated and distributed compute. But these are challenges, how we actually build robust software within AI data labs in Europe, in testbed centres, how we actually scale for the European health data space, which in 2029 in Europe, all health data, all clinical data will be available for research. So how do we scale to this? How do we build that software qualities metrics? We have quality at the moment that checks individual packages, but how can we check that the quality of the software is working very well within an AI workflow and a containerized system? And how can we build GPU native compute? The final thing I will mention is our funding is very much US-centered at the moment. We rely on NIH funding. We have had funding from Chan Zuckerberg. We've had various other sources of funding. How can we diversify our funding? How can we look to IHI programs? If you want to partner with us on an IHI program, we would be delighted to do that, because the software that we're building will actually underpin a lot of the European health data space innovation. Thank you.